Welcome to G Fun Facts Online. Today, we're diving deep into a silent, unceasing struggle that happens all around us, in our parks, our forests, and our backyards. It's the battle for water. For plants, rooted in place, there is no escape from drought. They can't run to find an oasis. Instead, they must stand their ground and fight. And to do this, they've evolved a stunningly sophisticated internal defense system, a molecular toolkit that allows them to perceive, react to, and survive the life-threatening challenge of dehydration. It's a microscopic symphony of signals and fortifications that puts most human engineering to shame. So how does a simple plant know it's thirsty, and what does it do about it? Let's start at the very beginning, with the first hint of trouble. A plant's ability to respond to drought begins with its power to feel the lack of water. This isn't a conscious feeling, but a physical and biochemical one. As the soil dries, the most immediate effect is a loss of what's called turgor pressure. Think of a balloon filled with water. Turgor pressure is the force of that water pushing against the balloon's skin, keeping it firm. In a plant cell, this pressure is crucial for keeping leaves and stems rigid. When water becomes scarce, this pressure drops. The cell loses its firmness, and the plant begins to wilt. This change in physical pressure is one of the very first alarms. The cell membrane itself can sense this compression, and it's thought that changes in the membrane's thickness or tension act as a direct sensor, kicking off the response. The cell wall, that rigid outer layer, also plays a key role. As the cell shrinks from water loss, the connection between the membrane and the wall becomes strained. Special proteins embedded in the membrane known as receptor-like kinases act like sentinels. They detect this strain and relay the message of dehydration to the cell's interior. This initial perception of stress is then amplified. Two key messengers are released: calcium ions and reactive oxygen species, or ROS. The stress of drying soil causes a sudden influx of calcium into the cell's cytoplasm. This calcium spike is a widespread alarm, activating a whole range of proteins. At the same time, drought stress leads to an increase in ROS. Now, high levels of ROS can be damaging, but in controlled amounts, they act as a critical alarm signal, essentially shouting, "Emergency! Emergency!" throughout the cell. Once these initial alarms have sounded, a master conductor takes the stage to orchestrate the plant's entire defense strategy. This conductor is a plant hormone, or phytohormone, called abscisic acid, better known as ABA. Under normal, well-watered conditions, ABA levels are kept low. But as soon as drought hits, its synthesis is rapidly ramped up, primarily in the roots and leaves. This surge of ABA becomes a systemic distress signal, traveling throughout the plant to initiate a vast spectrum of survival responses. The ABA signaling pathway works like a beautifully simple yet elegant molecular switch. It involves three core components: ABA receptors, a group of inhibitors called PP2Cs, and a class of activating proteins called SNRK2 kinases. Here's how it works. When the plant is happy and hydrated, the PP2C inhibitors are active. They keep the SNRK2 kinases suppressed, essentially holding the drought response button in the off position. But when drought strikes, ABA levels rise. These ABA molecules bind to their receptors. This binding changes the receptor shape, allowing it to grab onto and disable the PP2C inhibitors. With the inhibitors now neutralized, the SNRK2 kinases are set free. They become active and are now ready to execute the plant's defense plan. They are the key effectors that carry the ABA signal forward. So what's the first thing these activated SNRK2s do? They trigger one of the most critical and immediate responses: closing the stomata. Stomata are microscopic pores, usually on the underside of leaves. They are absolutely essential for taking in the carbon dioxide needed for photosynthesis. But they are also the primary gateways for water loss through a process called transpiration. When water is scarce, closing these gateways is a plant's top priority. The ABA signal, carried by the SNRK2 kinases, targets ion channels in the guard cells that flank each stomatal pore. This triggers a mass exodus of potassium ions from the guard cells. As the ions leave, water follows through osmosis. This causes the guard cells to lose their turgor pressure, become flaccid, and collapse inward, sealing the pore shut. This rapid response drastically reduces water loss from the leaf, buying the plant precious time to mount a more long-term defense. While closing the stomata is a quick fix, surviving a prolonged drought requires a fundamental shift in the plant's entire biology. This is achieved by a massive reprogramming of gene expression. The plant essentially rewrites its own operating manual for survival mode. This genetic response is controlled by a network of specialized proteins called transcription factors. Think of transcription factors as project managers. They bind to specific sections of DNA to turn entire sets of genes on or off. In the case of drought, the main managers working for ABA are from a family called AREBB/ABF. The activated SNRK2 kinases switch on these AREBB/ABF transcription factors. They then go and activate a huge number of protective genes. These genes produce things like osmoprotectants, which help cells hold onto water, detoxifying enzymes, and special protective proteins. Interestingly, plants also have another pathway that doesn't strictly depend on ABA. This pathway uses a different set of transcription factor managers called DREB/CBF. These are particularly important for responding to the osmotic stress caused by dehydration. While it's called an independent pathway, there's a lot of crosstalk between the two. They form a complex, integrated regulatory network that allows the plant to fine-tune its response to the exact severity and duration of the drought. So what are the actual tools and shields that these activated genes produce? What does this cellular army actually consist of? One of the most important groups of molecules produced are the late embryogenesis abundant proteins, or LEA proteins. As their name suggests, they were first discovered in plant embryos, right as the seed is undergoing extreme dehydration to enter a dormant state. It turns out, they are also produced in leaves and roots during drought. LEA proteins are like molecular bodyguards or chaperones. As water becomes scarce inside a cell, other vital proteins can start to misfold and clump together, which is disastrous. LEA proteins step in to prevent this, binding to other proteins and to cell membranes to stabilize them and protect them from damage. They are the cell's personal survival specialists. Another key component is the regulation of aquaporins. Aquaporins are channel proteins that act as water gates in cell membranes, controlling the flow of water in and out of cells. Their role in drought is a delicate balancing act. On one hand, they are vital for pulling water up from the soil through the roots. On the other hand, you don't want leaky gates that let precious water escape from cells. So, during a drought, the plant dynamically regulates them. Some aquaporins, particularly in the roots, might be ramped up to maximize water uptake from the drying soil, while others in the leaves might be shut down to prevent water loss. Beyond proteins, drought also triggers major metabolic changes. One of the most important is known as osmotic adjustment. The plant starts producing and accumulating high concentrations of small, soluble molecules, like the amino acid proline and various sugars. These are called compatible solutes or osmoprotectants. Their job is to make the cell's internal fluid more concentrated, which lowers its osmotic potential. This helps the cell to hold on to its existing water and maintain turgor pressure, even when the surrounding environment is dry. But drought causes another problem: oxidative stress. The disruption of photosynthesis can lead to the overproduction of those reactive oxygen species, or ROS, that we mentioned earlier. While they act as an initial alarm, in high quantities they are like ricocheting bullets, damaging proteins, lipids, and DNA. To fight this, plants ramp up their antioxidant defense systems. They produce enzymes like superoxide dismutase and catalase, along with antioxidants like vitamin C, which all work together to scavenge and neutralize the excess ROS, mitigating the damage. Perhaps one of the most incredible aspects of this entire process is that plants can remember a drought. A plant that has survived a period of water scarcity can respond more quickly and more effectively if it faces another drought in the future. This isn't a memory stored in a brain, but one encoded at the epigenetic level. Epigenetics refers to changes that don't alter the DNA sequence itself, but modify how it's used. Think of it like adding sticky notes or bookmarks to the pages of the plant's genetic instruction manual. During a drought, specific chemical tags, like DNA methylation or histone modifications, are placed on drought-responsive genes. While some of these tags are removed after the stress passes, others can persist for a long time. These persistent tags act as a molecular memory. They prime the defense genes, keeping them in a state of readiness. If a second drought occurs, these primed genes can be activated much faster and more strongly, boosting the plant's resilience. In some cases, these epigenetic marks can even be passed down to the next generation, a form of transgenerational stress memory. The cellular drought response is a testament to the remarkable adaptive power of life. It's a multi-layered, highly regulated system that uses an astonishing array of molecular tools. From the first physical sensation of wilting, to the hormonal emergency broadcast of ABA, to the deployment of a genetic and metabolic army of protective molecules, every part of the plant is recalibrated for one goal: survival. Understanding this intricate toolkit is more than just a fascinating piece of science. In a world facing the growing challenges of climate change and water scarcity, deciphering these secrets is critical for global food security. By harnessing this knowledge, scientists are working to develop crops that are more resilient to drought, capable of providing stable yields in a thirsty world. The silent, cellular symphony of the drought response holds a key to our future, teaching us profound lessons from the quiet wisdom of the plant kingdom.